Hi, I'm Dr. Shakthi, and if you're watching this video, you're probably here to understand more about fluidity of the membrane. Now, I already discussed about the structure of phospholipid and all that in another video. I put up the link over here if you want to. So once you understand that, we can go on to fluidity of the membrane. So we have a phospholipid structure here. We have a glycerol base over here and fatty acid chain. And there's a phosphate part with a head part attached to it. So this could be choline, this could be ethanolamine, serine. So it depends upon it. So the thing is, the phospholipid which makes up the membrane, this is the hydrophilic, this is the hydrophobic part, this is the extracellular fluid. Over here is a intracellular fluid. So this is a bilayer. Now over here between them, there's something called an interaction. Okay, so between them we got an interaction. Okay, now consider there is temperature. So when you talk about increasing temperature, it increases a lot of things, mobility as such, the Brownian movement, diffusion, everything. Okay, so when temperature increases, the chance for this to move around increases. How does this normally move? So this can move around laterally like this. This can hinge like this and move. This leaflet by itself can rotate like this. So these are various ways by which this can move. So this is something sort of a lateral diffusion. So the lipids along with all the proteins that are there, that can displace itself laterally in this hydrophobic area of the membrane. So that lateral displacement is over there. So this is how the mobility is there. Okay, so when the temperature increases, the thermal energy that is there increases, and that thermal energy makes it want to move. But how far can it move? That depends on the interaction energy. If the thermal energy is more than the interaction energy between them that is hindering the movement, then of course there will be mobility. So when a lot of mobility is there, this will exist in the fluid like what we call the sol stage. Okay, so it is in the sol state that we see it. The exact opposite of that, when the thermal energy is not enough to make it move over the interaction energy. So the interaction energy outweighs the thermal energy. So what is going to happen? There is not much mobility, it's more rigid structure, and that will go into a gel state. So that's how the membrane exists. So the temperature at which it can change from the sol to gel. Okay, so that temperature is your transition temperature. So at transition temperature, above it goes into your sol state, and below that it goes into your gel state. So once you understood this, let's go on to understand the things that affect your interaction energy. So as I mentioned already, interaction energy is there, that is a hindrance to your mobility. So what are the factors that affect interaction energy? One thing is the length of the fatty acid. So you can see over here this fatty acid chain and this fatty acid chain. There are more number of hydrocarbons in this fatty acid chain than this. Okay, so just imagine you take a really tall person and you take a very short person. Put them in a very confined area of the same space. Okay, the volume of that area, the volume of that space is the same. Okay, put a very tall person and put a very short person. Who will have more freedom to move around? Of course, it will be the short person. Okay, so in a similar way, the longer the fatty acid chain is, the more difficult it is. Okay, and shorter it is, more freely mobile it is. Okay, so that is the length of the fatty acid chain. Another thing is the presence of double bond. That makes it whether it is saturated or unsaturated. So what does the double bond in this structure do? So this is, as you can see, saturated. There is no double bonds exist over here. If double bonds exist, it will be like this. Can you see this over here is where you have the double bond. So once there is a double bond, there is a kinking of the structure. So this exists in a cis formation now. Okay, now the trans, it exists in a cis formation. When there is a double bond, there is a kinking. Okay, so let's take an example. You consider going to a movie theater or maybe even an aeroplane. Okay, so you're on the flight and there's another person. You got your seat and you see that the person next to you has already kept their arms over the armrest. So once that is done, that person has a lot of mobility already to move around. Isn't that right? But we who have come later, who could not do that, have little mobility. So that kinking kind of gives this a little more mobility. So presence of double bond and a shorter fatty acid chain are things that give increased mobility. Okay. On the other hand, less double bonds and longer fatty acid chain kind of increases the interaction energy, and the other things decrease the interaction energy. Now the next mystery when you talk about fluidity is cholesterol. So first thing is you have to understand what cholesterol is. So cholesterol comes in a lipid, but it's a little different from all the other lipids. First thing is, this is the structure of cholesterol. When you talk about cholesterol, you can see that you have a sterol head. Okay, so the sterol nucleus is there. On top of it, there's one part where there is a OH. So that part is going to be hydrophilic. Rest of the part, along with the hydrocarbon below, that is going to be your hydrophobic part. So this is also amphiphilic, right? Okay. Now what happens is, this structure is different from other phospholipid. It has different transition temperature. The temperature, it alters everything. So when you put a cholesterol over here, what it does is, it comes in between like this. This hydrophilic part over here comes between the heads of this. The sterol part is there and the hydrocarbon part is there. So when you talk about cholesterol, you can talk about with respect to the concentration of the cholesterol and the temperature. Two things I'm going to discuss now. First thing, we'll talk about your concentration. Yeah. So when you talk about the concentration, generally when the concentration is modest, not that high, what happens is, this sterol part, that is now going to go and hinder the movement, the mobility of your phospholipid hydrophobic part. So what does it do? It decreases the fluidity. So a modest concentration, what it does is, it decreases the mobility. But if the concentration is very high, so that everywhere it is there, not only here, 1, 2, 3, everywhere it is there, and it's like blocking everything. So although this does attach to it and give some sort of hindrance, it kind of acts as a blockade between the two phospholipid. So you understand what is happening? Now when the concentration is very high, it's actually acting as a barrier for the interaction that we are talking about earlier. So much so that now, because of that, this cholesterol is now going to increase fluidity, because the interaction between the phospholipids are reduced. Okay, so although, just remember, in case you have a doubt, although this does by itself cause a hindrance, high concentration of it, the hindrance that is caused is much less than the interaction energy that it helps remove. So ultimately, the fluidity increases. So that is regard to its concentration. Now when you talk about temperature, cholesterol, what it does is, it acts like a buffer. That's the first thing you have to understand. Cholesterol acts like a buffer. So when the transition temperature increases, it tries to bring it down. As in, now the temperature, but whatever is supposed to happen to the membrane. So what happens to the membrane when temperature increases? Thermal energy increases, fluidity increases. So it tends to go to the salt state. When transition temperature is, the temperature becomes over the transition temperature. Okay. So now at this stage, when it's supposed to go into salt and increase fluidity, this cholesterol, what it does is, as we had mentioned earlier, this sterol part will cause a hindrance and try to reduce the fluidity. So it acted as a buffer to reduce the increase in the fluidity. On the other hand, once the temperature goes below transition temperature, what cholesterol does is, again, the same thing that we had mentioned. So it is the in between. So when it goes down, the fluidity actually decreases. So when the fluidity decreases, suppose you go into a gel state, what it does is this hindrance between them is created, so the interaction energy is reduced, and cholesterol kind of tries to reduce the decrease in the fluidity that we just saw. So ultimately what it does is, when transition temperature increases, it kind of opposes the change for it to go into solid state, and when it goes below the transition temperature, it opposes the membrane going into your gel state. So it kind of acts like a buffer. All that is very dependent. How much cholesterol is there in the membrane, in case it needed. A lot of organisms can generally change the concentration of cholesterol in the membrane as and when required. Okay, so a lot of processes like this are there. So this is the basic thing that you need to understand about fluidity of membrane. Yeah, that is the interaction energy, the thermal energy, the length of the fatty acid, the double bond, and of course the cholesterol. So I hope you understood this. Thank you.